All right, so we're going to get started here. Sort of at the beginning, kind of back where the Earth is forming around 3.6 billion years ago. And after it forms, what we find is evidence that shows us that the first cells came about around 3.8 billion years ago. So we have this time period of a very long time, really, about 1 billion years, for the first cells to come about. So what we're going to look at right now is the ideas and the evidence on how that happened, or how that potentially happened, and what evidence we have, and what are some of the explanations. So, what do we know? First off, we have some evidence that shows us what the early Earth's atmosphere was like. So how would we know that? Volcanic activity of the Earth created rock that is very porous and can trap inside it little air pockets. And so people have for many years been analyzing that gas inside those rocks to come up with a combination of chemicals that were potentially in the atmosphere. Deep ice cores, also that would have happened after a while, but again, bubbles trapped in ice that's very, very, very old can give us samples of what the atmosphere was like. And so people can then age the rocks or age the ice, and over time they can start to put together a picture of what the atmosphere was like at some point in time and how it had changed from one time to another time. All right, so that's kind of the idea about how we, as today, get some of this evidence, right, where we find it. Is it exactly going to be like, was it exactly like that? We don't necessarily know, but we have a lot of evidence to suggest that it was. So what was in the atmosphere? Well, there was a lot of nitrogen and carbon dioxide. There was also ammonia, methane, so we got methane here, and ammonia, hydrogen sulfide, and just hydrogen. In time, what happened is condensation would occur as well, and water forms and starts to build our Earth's oceans. And so now some of these molecules from the atmosphere would be in the oceans, and the ocean is a big soup, right, of these sorts of molecules. We have some experiments that have taken place that can get at the first sort of hurdle in our sequence of events. So what we need to get to here are, cells. That's kind of the goal. And to get to cells from just molecules in the atmosphere, how would that happen? Again, so this is our explanation of that based on the evidence that we have. First, we have an abiotic synthesis of molecules. So we have non-biological molecules, and we need to make some biological molecules from them. So how could that potentially happen? Back in 1953, so this is obviously not on this time scale here, it's right up here basically today, even though 1953 will seem like a while ago for us. It was really nothing ago compared to this timeline we're talking about. Two scientists, Stanley Miller and I forget Yuri's first name, he was really the student who really did the main work, set out to do an experiment where they gathered up a number of these molecules, these gases, put them together, and then they gave it a spark of electricity, and they would see, so this to give some power, simulating, say, lightning from storms in the atmosphere. And what would they get? Well, they found amino acids would just form from these. And so if we look at it, we have molecules that are the basis of what is an amino acid. So first off, if you don't know what amino acid is, and you're like, I don't really see where this is coming from. So an amino acid is a central carbon. Okay. We have carbon dioxide and methane in the atmosphere. Methane here, see carbon with 4 bonds to it, 4 hydrogens. So in an amino acid, you have the amino group, then you have a carboxyl group, a CO, OH like this. Well, that's carbon dioxide is carbon double bound to 2 oxygens. Okay, so that's our CO2. Then an amino acid is an amino acid, so it has an amino functional group, but we have ammonia in the atmosphere, as well, so that's coming for this amino group. Hydrogens, there's typically always a hydrogen attached to this carbon here, and then in our amino acids we have 20 that are coded for in our DNA, and there are a number of other amino acids that we could find, and they all vary. And so they vary in what is attached here. So it could just be another hydrogen, it could be another carbon, it could be a carbon with the hydrogens like this, it could be a whole variety of other sorts of things, right? And that's what makes them all different. But you can see all the main components, the main elements that are needed to make amino acids were all here in the atmosphere. Some other people have ideas that would say, maybe, but some of the atmospheric gases are missing, especially concentrations of things like hydrogen sulfide, which is a reducing agent. And so we'd need this to have some of these things occur. What we do know is that deep in the oceans, we have areas called hydrothermal vents. I've been lucky enough to actually visit hydrothermal vents in a deep sea 3-man submersible along the Juan de Fuca Ridge off the Pacific coast of the United States. And in these vents, what essentially is happening here is that cracks in the Earth's crust, water from the ocean seeps down in, it is superheated, and then it shoots out other areas on the bottom of the ocean, and with it, it brings all sorts of minerals and chemicals from deep within the Earth. A lot of hydrogen sulfide is here. A lot of the organisms that live at these vents use sulfur as the electron acceptor instead of oxygen in their electron transport, and so they do things a little bit different. And so some people think, oh, because the early Earth wouldn't have had much oxygen in the atmosphere, that the hydrothermal vents may have been the place where this all happened. So where it all happened is still a matter of investigation. It may have happened most likely multiple places, really all at the same time, and some were more successful than others, and some gave rise to variations of sorts of things. But all these things can happen. We can simulate them in a lab, and we kind of know the atmosphere. So you should kind of know what were some of the main components of the early Earth's atmosphere. Nitrogen and ammonia. We have carbon dioxide and methane, hydrogen, and then hydrogen sulfide. Okay, so kind of keep those in mind. Then we have water forming into the oceans, and then we have the potential beginning of the first molecules. Right? Now that we have some molecules, and a variety of others, not just amino acids, but other sorts of molecules as well, how do we get macromolecules? So macromolecules are the proteins and the nucleotides of DNA and RNA and these other sorts of molecules, and carbohydrates and so forth. So we also can have lipids and things actually forming, from this structure, because lipids are really just long chains of carbons, and hydrogens that could come together. And so what we can find is that some experiments were done where macromolecular synthesis can occur spontaneously with some energy put into it, but essentially without the help of an enzyme, without the help of ribosomes. So we can actually go from single amino acids like the one I had here to chains. We have one amino acid joined to another amino acid, joined to another amino acid, joined to another amino acid. And so we're now starting to build a polymer or a polypeptide, which would be the beginnings of proteins, which have three-dimensional shape and carry out a lot of the activities within our cells. The same sorts of things could happen potentially for more complex molecules like this that I have up at the top here, which is, well, this is really a structure for ATP, but we have here ribose in the center, a sugar, a 5-carbon sugar. We have the nitrogenous base adenine, right? And then we have some phosphate functional groups added to it. This is also the core structure here of our DNA and RNA. Right? And so the interesting thing is a lot of molecules that we have that are used for a variety of different things actually have very similar or related structures. So it's not that millions of different variations do come about, but they're really based on sort of a common core structure, right? And certain specific molecules and elements like carbon, hydrogen, oxygen, nitrogen pretty much make up most all of our biological molecules. So we need those elements, which we do have here. And so we know that just adding some heat and adding in again some energy, kind of like in these other experiments, these molecules can form on their own. So definitely possible with the heat of the hydrothermal vents that some of these early molecules could have then come together and we start forming these. But these are just molecules. So it's just, again, a big soup of molecules. So we have, let's say we have amino acids, floating around in the oceans, and some sugars, and lipids, and the precursors to our DNA and RNA, so our nucleotides, which will give rise to the nucleic acids. How do they get put together into a structure that's going to be a cell? So our next step here is to form something called a protobiont. So for this, what we have to think about here is a little bit come to the lipids. All right. So one particular kind of lipid that you should be familiar with is a phospholipid. All right, a phospholipid is the basis of, it's a molecule that's the basis for all of our cell membranes, internal membranes and our overall cell membrane itself, the outer cell membranes. A phospholipid is essentially a glycerol that has 3 carbons like this. Oxygen's attached here, and then there's chains of carbons. I'm not going to draw all the detailed chemical structure, but you can see here, what do we need to make one? Well, we have all the carbon, oxygen, hydrogen, essentially. And then here we have phosphorus and then some polar molecule, meaning it's something that's very water soluble, right? So it has more oxygen, essentially, in it, or nitrogen are there that it can interact with water. As we simplify these sorts of structures, we find that there's a polar part that interacts with water, like this, and then there's these non-polar parts, which is just carbons and hydrogens, that doesn't interact with water. And there's 2 of these chains, which are referred to as tails, right, like this. So we have these 2 tails, of the phospholipid. Now, if you were to take a phospholipid, well, first off, just take an oil, like you're familiar with olive oil. You take olive oil, you mix it with water, shake it up. They mix a little bit for a few seconds and they just immediately start to separate. That's because the olive oil, instead of having the polar group, also has another chain, and so it has no real polar parts, it just completely separates. The phospholipid does have a polar part, so it will start to interact with water a little bit. So if you did the same thing with a phospholipid, which would look like an oil, and then you mixed it with water, it would form a layer, just like olive oil would. Then if you shook it up, what would happen is, as they would mix, it would seem to vanish and be like, oh, it actually dissolved, but didn't really dissolve. What would have happened is it would have formed 2 sorts of structures. One is a more simple structure called a micelle. So a micelle has the polar heads oriented to the outside and the nonpolar tails oriented toward the inside. So there's no water in the center, but there can be water out here. And we can find these naturally can occur, right, in the oceans. We can just form these micelles just from wave action, even along the rocks, along the beach, and then the lipids can actually form into these little structures. In addition to that, we could form some slightly more complex structures where we start to get layers of phospholipids arranging themselves. So you have to bear with me for a second. So we have the one layer, just like the micelle here, right? We have, these are the polar heads, the parts that interact with water. They're out on the outside so they can interact with water. Draw the tails here, a lot of tails to draw, okay? And then you have a second layer. So we have another phospholipid bilayer. And this would be 3-dimensional. Remember, this is a sphere, so you have to kind of keep that in mind. So it's a ball, essentially, with a hollow core, except the core isn't completely hollow. Inside the core is water, okay? And so this is a structure called a liposome. And again, these can just naturally occur. We can just form liposomes as well just by agitating the phospholipids. So now what we're getting is this idea that, and I'm going to get rid of our atmosphere because now we're down into the water, all right, and starting to get to the protobionts and our first cells, which is where we're going to end, and then we'll start up with another lecture once we get there. All right, so liposomes form, and inside the liposomes are some of these molecules, right? So now we have, a membrane, a protobiont. So it's kind of bio meaning life, proto meaning kind of before. So it's something that's, it's not really a living cell yet, right? But inside it, we could have amino acids, sugars, other lipids, and other nucleotides. And these molecules can interact with one another in a variety of different ways, some meaningless sorts of ways, creating sort of random stuff. And others could be more meaningful, or something that's more long-lasting, right, in this case. What we have is this other than idea, which we refer to as the Rna world. And it's sort of at this protobiont stage is where the molecules can actually start to interact with each other a lot more because they're in close proximity. When they're just free in the oceans, it's a lot less likely that they'll be bumping into each other. Just like if there were like 2 people in a football stadium and the rest of it was just empty, the idea that you just bump into each other would be rare. But if there were thousands of people, the idea that you'd bump into each other is going to be great. So you get a lot of molecules in close proximity. They're going to bump into each other, which means they'll react with one another and they might form new molecules. And then some of those molecules may do something, right, inside that structure. And the idea is that with billions and trillions of different types of these protobiont structures forming with all sorts of different combinations of molecules inside them, most of them leading to nothing, that some of them led to something that would be the precursor to our first cells. Those, we believe, have formation of RNA in it. So again, so now RNA is ribonucleic acid, okay? So the R of RNA is ribose, all right? So that's this sugar here. This is the sugar ribose. It's a nucleic acid because then it also has the nitrogenous base, right, which would have to be also manufactured from nitrogen compounds, also some carbons, in here, and then these phosphates are where the energy comes from, holds them together. It's where the bonds form that actually join the different nucleotides together, which is a sort of a separate topic, but that's the basis of the structure. We have RNA in our cells. All living things have RNA. Some organisms only have RNA. So a virus, there are some viruses, for example, that are RNA viruses. They have no DNA whatsoever. They only have RNA. But for us, RNA is sort of the intermediate between DNA and protein. And in between is RNA. And for us, this is a process we refer to called gene expression, with the idea being that the purpose of the DNA is to carry information. The purpose of the proteins is that they carry out actions inside the cell. They're enzymes, they're active transport pumps, they're structural molecules, they're motor proteins, they move things around, so they're doing everything that's carrying out life. And the RNA for us is the in-between. The interesting thing about RNA is that it can actually do both jobs, okay? So RNA is nucleotides and DNA is nucleotides. So here I have the A and I have UGC. Well, DNA is ATGC. So the information, the code, the messages carried by the DNA are really in these nitrogenous bases, and RNA has the same ones, or in one case, just a slightly different one, the uracil versus thymine. So carrying the same information, RNA does as DNA does. DNA, which is just sort of a static molecule that's just folded into a very regular structure, is unlike RNA. RNA is single-stranded and can be folded into very complex structures. Okay? RNA can actually carry out chemical reactions like proteins. So Thomas Ketch and discovered that, so its name is C-E-C-H, yep, I'm doing a spelling. Discovered that RNA can act as ribozymes, meaning that the RNA molecules can be folded into structures that can then carry out chemical reactions. Some of those reactions is that they can copy themselves, which is significant because this means self-replication. So that means that some of these protobionts that then formed RNA within them, some of those may have had RNA that formed ribozymes, and that the ribozymes could then make copies of the other pieces of RNA, and then this structure could split into additional structures, and that the material inside the structure could have been replicated or duplicated before that they split, and then the information is carried on to the next generation or next group of cells, and so these cells then carry on. So what we start to have as we get these self-replicating molecules, which is RNA, that natural selection is going to occur on them. Meaning, again, some of these things, some of the original protobionts lead to nothing. They have just disorganized bunches of molecules within them that don't really react or do anything. Some of them react and they don't carry out anything positive. But again, after hundreds of billions and trillions of possibilities, that some of them do actually form something that has a reaction, and that those reactions carry on. So we can actually again see some of these things in a lab. We can actually see that some RNA over time can be selected for, that the RNA in a tube can have different versions that do a little bit of ribozyme activity and actually can cut DNA or copy pieces of RNA or carry out different types of jobs, and that if they're selected for, and you take some of those and put them into another generation, let them replicate themselves, there'll be more of them that do a better job, and then you can select some of those and put them in another generation, and some of them will do even better job than the ones before, and you can artificially in a lab observe RNA just becoming much more effective and efficient. So it is likely that in nature, over time, over a very long period of time, billion years, that this most likely could have happened. And what this would have given rise to was a membranous structure with unique molecules inside them, some of them being RNA, which can carry out chemical reactions and also store information that could be passed on to other generations. So this is then the beginning of something that is like a cell, right, that would become an actual living cell. And so that's kind of what we'll end with. And so the RNA world is this hypothesis that RNA really came before DNA and proteins. There's, again, a bunch of evidence from modern biology. There's some organisms like some virus have there are some RNA viruses, okay, so their genome is only RNA, and what they have is a specific enzyme called reverse transcriptase. And what that allows is the organism's genome, which is RNA, to be used as a template to make DNA. So that DNA can come from RNA. So we said normally in our sort of central dogma that RNA comes from DNA, which is generally the case in most living things, but in this RNA world scenario, we would have RNA first, and we have to get DNA from RNA, and we can see examples today that enzymes do exist that can do that. Some organisms have that and can actually do it. So the idea that, again, it came about at some point in time, well, it had to come about at some point in time, and that's partly one of the ideas to explain where these organisms originated from. So so what do you need to know from this? Again, the atmosphere, the basics of the structures, the liposome structure, the protobiont concept, that it's a structure that would have these molecules in it, the RNA world theme, that you're going to have the RNA as a self-replicating molecule that can carry on information and carry out cellular activities. That's the phospholipids that are forming into these liposome structures. Some Miller and Urey's experiment back in 1953 a little bit, Thomas Cech kind of showing the ribozymes. Those are some of the main takeaways then from this lecture. So the next thing we'll be doing is getting into first cells, prokaryotic cells, and looking at some characteristics of bacteria and archaea.